Good morning, everyone. My name is Jeff Morby. I'm co-chairman of the Cure Alzheimer's Fund. And today, the purpose of this meeting is to have Dr. Rudy Tanzi provide us with an update as to the status of Alzheimer's research. Rudy is one of the outstanding researchers in the world. I'll give you a little bit of his background in a minute. But before I introduce Rudy, I'd like to say a couple things. First, in 2004, 3 families established the Cure Alzheimer's Fund. And our objective at that time was to find a cure or preventative for Alzheimer's disease as fast as we could. And we had the good luck of running into Rudy Tanzi, who was known to be the top geneticist in the world in the Alzheimer's field. We made a deal with Rudy. The deal was very simple. Rudy was to put together the finest group of scientists in the world in Alzheimer's research, who together would develop a research strategy designed to find a cure for the disease, and we would fund Rudy. 17 years later, that's exactly what has happened. We now have more than 90 world-class researchers around the world doing research on Alzheimer's. We funded about $150 million of research. We're funding about $20 million a year. And Rudy has put together this marvelous team that is really breaking all kinds of bounds of knowledge in the area of research. At the outset, when we first started this, we made a fundamental decision, which was very, very good. We decided to focus on the genetics of Alzheimer's. To really understand how to cure a disease, you really have to understand how the genetics works. And that was our focus at the beginning. And over the last 17 years, we've done 3 major genomic scans, meaning we have brought together these large databases of people with Alzheimer's disease. And that allows us to do cross correlations among these people to see what diseases actually are caused by different genetic defects. And we now have one of the largest databases in the world of the genomics of Alzheimer's. And we are the leader in this area. And Rudy has been the guy that has put this all together. So we are making great progress. Rudy's going to talk to you about that. So I won't say anything more, except I want to then go in to introduce some of our other friends here that are on the screen. First of all, I want to mention that we have with us Bruce Ekman. And the reason Bruce is there is Bruce, among many other things, runs the speakers program in Martha's Vineyard, and also cooperates with, that's the speakers program for men, and he cooperates with the speakers program for women. And I had made a presentation several years ago, and they've really been wanting to have a follow-on presentation. So he's been a prime mover in getting this going. So Bruce, I'd like to have you say a couple words. Go ahead, Bruce.
Thank you, Jeff. 25 years ago, approximately, Stanley Snyder, who is a member of our group on Martha's Vineyard, started a men's group. The idea of that was to have a place for men to get together and foster communication and fellowship with other men. And Jeff has been part of this for a number of years. And as he's mentioned, he made a presentation several years back. Typically, we either do a presentation out of personal interest, or we present a topic of which we are an expert ourselves. This is an example today of a member expertise coming back to play us back several years later. There is, as Jeff mentioned, a women's group as well that offers a similar platform, and we've been going on weekly over the summer for the last 25 years. Thanks for including us.
Thank you, Bruce. Now, secondly, I want to introduce Meg Smith. Meg Smith is really the center post of our interfaces with the scientists, because it's through Meg that we make the decisions to fund or not fund research. Meg is a brilliant person. She's a graduate of Harvard Law School. She's a former McKinsey consultant, and she probably knows as much as anybody in the world right now about Alzheimer's disease. She's going to be our host in the Q&A, and I'd like her to give you a little summary of what she plans to do. Meg?
Good morning, everyone. And Jeff, thank you so much for the very generous introduction. We are grateful to all of you for joining us today, and I will be representing you during the Q&A. I'll tell you about that in just a minute. But first, I did want to say that I have the privilege in my role as Executive Vice President of Research Management of working very closely with Dr. Tanzi and the other members of our research leadership group, who are all active researchers on the cutting edge of Alzheimer's science. They make all of our scientific decisions, and they set our priorities. And it is a true privilege to hear from Dr. Tanzi this morning. After he speaks, we will have a Q&A. During his talk, please feel free to put your questions in the chat section of Zoom. If you want the whole group to be able to see your question, you can just post it directly in the chat. If you would like for only me to be able to see your question, please direct it in the chat only to me. You'll see my name as Cural's Meg Smith. I will then take all of your questions and pose them to Dr. Tanzi after his talk.
And you'll also amplify also.
Yes, I'll combine and I will build on your questions, because I know we're going to have great questions from all of you.
Yep. Okay, with that, I'd like to introduce Rudy. I have his resume in front of me. It goes page after page. I'm just going to read the first part of it. Dr. Rudy Tanzi is Vice Chairman of Neurology and Director of the Genetics and Aging Research Unit, Co-Director of the Henry and Allison McCann Center of Brain Health, and Co-Director of the Mass General Institute of Neurodegenerative Disease at Mass General Hospital. He also serves as Joseph P. and Rosa F. Kennedy Professor of Neurology at Harvard Medical School. He received his bachelor's in microbiology and a BA in history, University of Rochester, and his PhD neurobiology at Harvard Medical School. He's famous, and not only that, he's a nice guy, and he likes to play basketball. So Rudy, would you please take over?
Great. Thanks, Jeff. I wish my knees still allowed me to play basketball. I've had to hang up the basketball shoes, just play out in my driveway with my daughter Lila now, but that's good enough. So thanks, Jeff and Jackie and to the group for having me speak today. It's great. This is, it's great, particularly today. September is World Alzheimer's Month. Yesterday was World Alzheimer's Day. I've been pretty busy with doing some media stuff for World Alzheimer's Day, getting the message out. It's an optimistic message. What I want to share with you today, that I think we've made great progress in understanding how Alzheimer's disease works and how best to stop it. And Jeff made the point that Cure Alzheimer's Fund begins with genetics. It's our foundation. We have over 100 Alzheimer genes now. The first 4 Alzheimer genes we found in the 80s and 90s. I found the first one when I was a student at Harvard doing my doctoral thesis. That's the amyloid gene. And what I'm going to tell you is that because we now understand the course of the disease, which is about a 20 to 30 year course, right? It's very similar to heart disease and other diseases that take years, if not decades, for pathology to accumulate before you see symptoms. Because we understand that, what I'm going to tell you is that treating the right pathology at the right time is how we'll both nip this disease in the bud and prevent it in the future. But it also tells us what we need to do to try to help patients right now. And we're doing both of those things. I want to be clear that stopping Alzheimer's, it's a bifurcated pathway. There's one thing you need to do. There's a few things you need to do to prevent it, which are different than how you're going to treat it. And a lot of how we prevent it has to do with how we preserve and promote brain health. So I'll be talking about that topic today as well. I want to mention Cure Alzheimer's Fund a lot during this talk because Cure Alzheimer's Fund, it can't be exaggerated enough that they have made just a huge difference in our understanding of the field due to the research that they've funded and the impact that Cure Alzheimer's Fund has had on the field. Paradigm shift after paradigm shift, seminal discovery after seminal discovery, it's just been amazing over the last 17 years now, just how many huge studies, big papers in the top journals will come out solely due to the researchers of Cure Alzheimer's Fund. I like to say the bang for the buck compared to other groups that fund Alzheimer's research is unmatched in terms of what Cure Alzheimer's Fund is doing. And speaking on behalf of all of the scientists that Cure Alzheimer's Fund funds, we are so grateful for the board and the Cure Alzheimer's Fund folks for believing in us and believing in some of the risks we want to take and the high reward, high risk research that's really paid off. And I'm going to tell you a lot about a lot of it over the next, I guess, 45 minutes or so. So let me, as pure Harvard and Mass General rules, I have to show my disclosures. These are over the last 5 years. These are companies in which I've either had a financial interest or have consulted for. So I need to just show that slide first. Now, I want to start with just something very simple, which is every year you go to your doctor for your physical. Think about that word. We call it checkup, we call it a physical. We call it a physical because you don't get a mental, right? They might talk to you and ask you, how do you feel? You get your heart rate tested, you get your blood pressure tested. But when you get from the neck up, like Patrick Kennedy once said during a talk we gave together, there's no checkup from the neck up. When you get above the neck, they're happy just looking at those holes in your head, up your nose, through your ears, in your eyes, and your mouth. They don't really talk a lot about that 3 pounds of gel in your skull called your brain, which is the most important organ in your body. And that's really the reason why neurodegenerative diseases like Alzheimer's and Parkinson's are probably the biggest challenge we have to healthy aging. Bottom line is that our lifespan has greatly surpassed our health span, but especially in the brain. The health span of our brain is not even remotely keeping up with our lifespan at this point. Modern medicine has us all living longer and longer, but our brains, we don't take care of our brains. And that's really the elephant in the room. Bottom line, we don't diagnose diseases like Alzheimer's until the brain has degenerated to the point of dysfunction. Imagine if we did the same thing in diabetes. Imagine if we said, we're going to wait till you lose half of your beta cells in your pancreas, and then we're going to treat you for diabetes. Or we're going to wait till you need a coronary bypass, and then we'll treat you for heart disease. Well, folks, that's what we do. That's what we do with Alzheimer's disease. And I'm sure that decades from now, or at least by next century, they'll look back in horror and astonishment at how we just have ignored the brain as part of just normal healthcare. We need to put brain care into healthcare. We need to know that Alzheimer's is beginning, because as I'll tell you, it starts decades before there are any symptoms. And that's when you need to hit it, just like we do with other age-related diseases, diabetes, heart disease, cancer, et cetera. For those diseases, for heart health, cancer, there's primary care. We start looking for we pay attention to early tumors. We do mammographies, PSA for cancer. We check cholesterol, blood pressure for heart health. All of this is routine healthcare. When you get to brain health, what do? Nothing at all. Maybe your physician might give you a MOCA, which is one of the cognitive exams. which really tells you very little. We wait till your brain degenerates and then we say, okay, now you're sick. And then we're reactive, we try to bring it back. We need to make treatment of Alzheimer's, Parkinson's, neurodegenerative disease proactive. And here's why. Alzheimer's right now is the most common form of dementia in the elderly. People ask the difference between Alzheimer's and dementia. Well, Alzheimer's is a type of dementia. If we say fruit and an apple, apple is to fruit as Alzheimer's is to dementia. It's the most common. I'm not sure if the apple is the most common fruit, but I just said that. There are almost 6 million patients in the US right now, costing over $300 billion per year. But if you look at how many people in this country have the pathology of Alzheimer's brewing in their brains without symptoms yet, it's probably close to 40 million. And they're not being detected, they're not being treated. Risk factors include, of course, age and family history, head injury, and sex, so gender. Women make up 2/3 of patients. Most caregivers are women. This is very much a woman's disease. But here's the scary thing, if that's not scary enough. The fastest growing segment of our population is 85 plus. If you look here on the side, here is the breakdown of age groups in 1960. There's this pyramid-like structure. By 2060, it will be a pillar. The 85 pluses are catching up, over 80 catching up. And the fact is this, current lifespan is 80 years old. After 85, based on epidemiology studies, 30 to 50% of folks are starting the symptoms of Alzheimer's disease. This disease just strikes half our population roughly by the time you reach 85. And right now our lifespan is 80. So we have a huge epidemic. This is a bigger pandemic, and cases will triple by 2050. Cure Alzheimer's Fund, you heard from Jeff about how it started. I won't go into that again, but it's an amazing foundation funding the top research. I'm so impressed that the board and certain donors pay the overhead. So when a dollar goes to Cure Alzheimer's Fund, it's a dollar back out. And they're the only Alzheimer's philanthropy with a 4-star rating from Charity Navigator and a platinum rating with GuideStar. So, but the main thing is Cure Alzheimer's is funding the highest impact work that's making a difference more than any foundation, even more than the NIH in terms of bang for the buck. And we couldn't be more grateful for their existence and for their support. So let's get into Alzheimer's. And one of the first things people ask me is, what's the difference between having a senior moment, there's actually a game called senior moments, and Alzheimer's disease? And it's a good question, because sometimes senior moments mean you're just not paying attention. And sometimes senior moments might mean that you're beginning Alzheimer's disease. And it's really tough to tell the difference, to be very honest. Part of the reasons for senior moments are illustrated in this slide. A child is very passionate about life, very engaged, very curious, very excited, wow moments all the time. You get older, we like to say you go from twinkle to wrinkle, right? That twinkle in the eye is often gone. You've seen it all. You become jaded. Nothing's exciting anymore. Wow moments become so what moments. Yeah, been there, done that. And so to a certain extent, with age, we just tend to become too busy, jaded. We pay less attention to what's going on around us. We're less interested. Of course, that can be fixed. That can be fixed with just mindfulness and being in the moment. But when these memory lapses start to occur so frequently and intensely that they challenge your quality of life, okay? So it's not just forgetting where you put your keys. It's not just forgetting that you had the toaster going, when you picked up a phone call. When it actually starts becoming the fact that you can't operate correctly, you can't budget, you can't balance your checkbook. You're in a town you drive all the time, or in your neighborhood, and suddenly for some reason you don't know how to get back home. When things like this start happening, this is when you have to think maybe it's time to see a doc. I got to tell you, there's a new program called mybrainguide.org that I helped develop. Mybrainguide.org is a website. And you can go on there confidentially, because some people don't want to talk about Alzheimer's with their docs. Some docs don't want to talk about Alzheimer's. Growing up, we called cancer the C word. No one wanted to talk about it. Alzheimer's is the A word. Well, mybrainguide.org, it's an app or there's, I'll show you the website later and the phone number, but you can just have a short test. And it's not a substitute for a diagnosis, but you do a short test privately. On the phone call, it's a robotic voice. No one knows. On the app, it's private. So you just get an idea of whether you should be seeing a doctor. And I think this is the type of thing we need to start doing, is destigmatizing Alzheimer's, because you're not going to be able to get help if you just wait to have full symptoms. We can try to help then, but it's much better to try to hit this thing early detection, early intervention, which I'll talk a lot about today. So what happens in Alzheimer's? Well, first we have to think about how our brains process information, cognitive processing. We live in a part of the brain right here in the middle of the brain called the hippocampus. Hippocampus is Greek for seahorse. And you can see it actually looks a lot down here like a seahorse. So what's happening is that from when you're a baby, sensory signals hit your eyes and your ears. And at first they're just signals, right? You can't make sense of them. A baby sees a red shoe. There's something bright, it has a certain shape. The baby doesn't say, oh, that's a red shoe, it probably belongs to my mom. But over time you learn, because your brain takes that color red, takes the shape of the shoe, takes that signal, and then converts it into meaningful information. And that happens in your hippocampus. And now that information is stored as memory. So the next time you see a red shoe, you remember that that's a shoe that your mom might own or might wear. Now what happens is in cognitive processing, in terms of just keeping track of the day in your hippocampus where you live, you might have a lack of attention. You're not paying attention, like I said earlier. That can lead to senior moments, just not being engaged, right? But on the other hand, there could be brain pathology brewing in your brain that's interfering with those 100 billion neurons and the trillions of connections they make, called synapses, that create a neural network that bring you your world. That neural network is being modified all the time. Neuroplasticity, everything you see is modifying your synapses in your neural network all the time, and modifying how you view the world and how you remember different events. Well, what Alzheimer's does is, by destroying those synapses and tearing apart the neural network, beginning in the hippocampus, which is the area where you process the world, that tapestry you've created of your world, every choice you've made, every experience you've had, it's shaping who you are, how you know yourself, how others know you. That tapestry is slowly pulled apart, thread by thread. And that's what we have to stop. We have to know when those threads are being torn apart and keep that tapestry intact. And I'm going to talk to you about how we're going to do that pharmacologically with drugs and pharmaceutical products. I'm going to talk to you at the end how we can do it with lifestyle, and what's the best way to try to preserve our brains with lifestyle. But first, let's talk about this pathology. The pathology include the amyloid plaques. That's these big sticky material here. You see the plaques here in brown. That they sit outside the nerve cells. And then the plaques, when they interact with the nerve cells, cause what are called tangles, neurofibrillary tangles. This is a protein called tau, which is normally making up part of your structure of your nerve cells. And that tau protein becomes sticky inside the nerve cell and makes these twisted filaments called tangles. So the plaques lead to the tangles. And then as nerve cells start to die with tangles, that triggers in the brain neuroinflammation. And that is when 10 to 100 times more nerve cells die when neuroinflammation ensues. And that's the pathway, basic pathway of the disease. Now, there's bad news and good news. The bad news is we all start to get some amyloid plaques and tangles after 40, certainly after 50. The good news in terms of how we can try to treat this disease is that it turns out you can have lots of plaques and tangles, but if you don't have in the brain that cytokine storm, I'm sure some of you know that term now, in neuroinflammation, you don't get the disease. So think about COVID. The COVID virus itself doesn't kill you. The COVID virus causes a cytokine storm. These are the chemical mediators of inflammation. And it's the inflammation where the body's trying to fight the COVID virus. The inflammation in the lungs and in the blood vessels, if it's too rampant, can lead to severe health consequences and death. Well, Alzheimer's is kind of similar, except it's not a COVID virus. Plaques and tangles are forming with age. Nerve cells are dying for that reason, but the brain reacts with a cytokine storm, and inflammation, again, meant to protect the brain. See, the brain's really primitive. It has a very primitive immune system. If nerve cells die for any reason, the brain assumes it's an infection and starts wiping out that part of the brain to protect the other parts of the brain. That's what neuroinflammation is intended to do. So we know from resilient brains, you can have lots of plaques, lots of tangles, just like Alzheimer's. But if you don't have neuroinflammation activated or induced by those plaques and tangles and that little bit of cell death that comes with it, the nerve cells survive and you don't get symptoms. So what I'm going to tell you, just to give you a head, kind of a head start, is to prevent this disease, we need to hit plaques and tangles decades before symptoms, and even inflammation. But when someone has the disease, it's not enough to hit the plaques and tangles. You have to put out the fire. You have to stop the neuroinflammation and protect those nerve cells from dying from that neuroinflammatory onslaught, just like what happens in COVID when there's inflammation in the lungs and the vessels and other organs. So why do we think amyloid triggers this disease? Well, it's the genetics. Back in the 80s and 90s, we found the first 3 Alzheimer genes. And I say we, I mean, it's always a team. I always like to say I co-discovered these genes rather than discovered. It's always a team effort. The amyloid precursor protein, that's the protein that gets chopped up to make the amyloid. The presenilins, those are the enzymes that chop up the amyloid precursor to make amyloid. Over 200 mutations in those 3 genes guarantee early onset familial Alzheimer's under 60 years old. Lifestyle doesn't matter here. This is only 2 to 5% of Alzheimer's disease, but it's the most severe form. And all of these 3 genes lead to more amyloid in the brain. And it's due to the buildup of this small protein called the amyloid beta protein, or we abbreviate it A beta. And it comes from this larger protein called APP, amyloid precursor protein. And then one of the most common risk factor for late onset Alzheimer's is ApoE, the ApoE epsilon 4 variant. If you inherit 1 copy of this, and that's 20% of the population, you have a 3.7-fold increased risk for Alzheimer's, usually in your 70s or 80s. If you have 2 copies of E4, you have a 14-fold increased risk in the 60s or 70s. But importantly, carrying an ApoE4 variant does not guarantee the disease. These mutations guarantee. And what ApoE does is it also affects amyloid deposition in the brain, but more at the level of how much you clear out. The mutations that guarantee early onset Alzheimer's have more to do with how you make the amyloid beta protein. And most of the mutations, you make a longer form of A beta, 42 versus 40. And that's what seeds the amyloid deposition. So we've learned something about how to translate genetics to course of disease from age-related diseases. And that is that, and this is something you may not have heard before. I don't even think Jeff heard this before, and Jeff's heard it all. The early onset mutations that guarantee early onset disease, usually tell you about the events that happen way before symptoms that begin the disease. So take heart disease, right? Cholesterol buildup that's going to lead to heart disease at older age begins decades before you're going to get coronary artery disease or even congestive heart failure. Well, guess what? How do we learn cholesterol triggers heart disease? Well, Brown and Goldstein, who won the Nobel Prize, had a rare family mutation. They had a family with a rare mutation that increased their cholesterol. And they said, maybe cholesterol causes heart disease. And people didn't really believe them. Well, what do we do in Alzheimer's? We had rare mutations in families that increased amyloid. And we say, maybe amyloid causes Alzheimer's. And that's still controversial. But now we know that with cholesterol and heart disease, you can't wait till you need a bypass to take Lipitor, right? You need to start managing your cholesterol decades before symptoms. What I'm telling you, the first take-home point today is it's exactly the same for amyloid. Amyloid, the genetics is telling us amyloid's coming before symptoms. You have to hit it before symptoms, just like we hit cholesterol before you get heart disease. And so that's what the first take-home point, and I'll get back to that. Now, as it turns out, and this is not surprising based on what I just told you, every trial that tried to bring down amyloid in the brain has failed, with the exception being Biogen's new approval of Aduhelm or Aducanumab. And that's a mixed bag of results. I'll get to that a little later. But why have all these amyloid trials failed? Even Aduhelm, one trial failed, the other trial barely worked in terms of some improvement in cognition. They failed because if you look here on the left, see this red line? This is the buildup of amyloid. This is 15 to 20 years before symptoms, the amyloid's building up. 15 to 20 years before. Think about cholesterol and heart disease. It's building up, it's coming up, it's coming up. And this dashed line in the middle here is when the first earliest signs of cognitive impairment appear. Well, by the time there's cognitive impairment, amyloid's already peaked and starts to even, some cases, go up a little bit. Or on this graph, another graph of it, in the orange here, it starts to even plateau, right? In this graph, which is a newer one from Randy Bateman on the right, it's 30 years before symptoms, the amyloid begins. If you see, look here. Originally, we said 15 to 20 years. So somewhere between 10, 15, 20, 30 years in different people, amyloid's beginning. And as amyloid accumulates, the tangles also start to form. You can see in green here, the CSF tau. And the tangles start killing the neurons. And then that death triggers the neuroinflammation and widespread cell death and dementia. Well, what this says is we have to hit amyloid early, okay? This is how it works. Amyloid, up to 30 years before symptoms, is triggering the tangles. I just got a message that says my internet connection is unstable. Can you hear me okay? Just want to do a check. Can you all hear me okay? Okay, thank you. So amyloid, way before symptoms, is triggering these tangles. And then as the tangles form, that triggers neuroinflammation. Now you can get tangles in other ways. You can have bangs to the head, boxing, football, rugby. You can have genetic triggers of different types of tangles where you don't need amyloid to get there. Alzheimer's is the most common dementia. And it's a disease where tangles form and start killing nerve cells because they're triggered by amyloid. But in less common dementias, including frontal lobe dementia, progressive supranuclear palsy, chronic traumatic encephalopathy, in the case of athletes who get bangs to the head, there are other triggers to get you to tangles without the need for amyloid. Okay, so Alzheimer's is the most common. We call tangle diseases tauopathies because tangles are made of tau. And Alzheimer's is the most common tauopathy induced by amyloid, but it's not until you trigger neuroinflammation that you get enough cell death to get symptoms of dementia. So that means we need to detect these things early. And now we have imaging. We have brain imaging where we can see plaques and tangles in the brain. These are FDA-approved protocols for doing imaging. They're not covered by healthcare because health insurance says, well, if you know you have amyloid and tangles in your brain, it's not actionable. There's nothing we can do about it in their mind. So they don't cover the test. So if someone wants to have a test for plaques and tangles, they'd have to pay roughly $5,000 per test out of pocket. But now there's also blood tests. There is a blood test for amyloid that actually does pretty well at predicting what's happening in your brain. There's a company called C2N in St. Louis that has a amyloid test that's very sophisticated. Out of pocket, that's about $1,400, but it's a pretty good surrogate for telling you what's going on in the brain if you're not having imaging. We have other blood tests for tangles. We have blood tests for whether neurons are dying, neurofilament light, blood tests for whether you have neuroinflammation going. And I'll talk about this other new blood test we just developed in my lab in a minute. We also have digital technologies. I'm involved, I started a company years ago called React Neuro, where we have a VR, virtual reality device, and it's doing a neuropsychiatric exam in a virtual reality format, 3D, which gives you a lot more information. It's scanning your eyes. You know, think about after you get banged in the head, or you think you have a concussion, the neurologist says, follow my finger. Well, in this case, it's following the eyes at nanosecond resolution. So it's because as inflammation occurs in the brain, gradually and insidiously with age, you can start to see changes in how the eyes track. This is much more acute if you get a bang in the head. I work with the New England Patriots, and I'm a brain health advisor for the Patriots and Belichick. If you get a bang to the head on the field, it's so acute. There's so much instant damage that the eyes tracking, you can see the abnormality just by saying, follow my finger. But this is a way to do it more in a more sophisticated way. So we're using this now we're using this device to baseline the Patriots, the Celtics, the Bruins to see where they are at the beginning of the season. When they get bangs in the head, we see where they go. We're also using it in dozens of senior living centers now, tracking residents to see how the brain health is doing. So this type of digital technology is now happening. Now, one of the newest blood tests is really interesting, and I know this looks a little complicated, but let me walk you through it. This is the Harvard Aging Brain Study being run by Risa Sperling. We just published this paper, Cure Alzheimer's Fund, and funded it together with the McCann Center. And what in this case, there are 300 normal older adults. They don't have Alzheimer's. And we're tracking them from everything from doing imaging for plaques and tangles. We're checking their blood for amyloid and tangles. We're doing serum biomarkers, urine biomarkers, just everything possible. We're saying, what indicators are there? What biomarkers are there that will predict how you do cognitively? And in this case, it's 6 years of tracking the same people doing this intensive type of analysis. And what you see here, if you look on the left graph, is the dotted line is going downhill with cognition in people who have amyloid in their brain. So the average person who has amyloid in their brain, based on brain imaging, you can see how they go downhill, right? Now, if you look at the blue line, I mean the solid red line here, these people have the same amount of amyloid in their brain, but look, they're hardly going downhill over 6 years. And what we figured out is because they have high levels in their blood of a cytokine called interleukin 12. And this was really surprising, very counterintuitive, because we thought having a cytokine that causes inflammation in the body, in the blood, would make things worse. So we said, how the heck does one of the pro-inflammatory cytokines, part of that cytokine storm of COVID, help? High interleukin 12, that's the cognitive trajectory versus this. And then there was another one called interferon gamma, another pro-inflammatory cytokine in the blood. The red here shows how people go downhill normally. But if you have high interferon gamma, look at this in blue, you don't go downhill. It even goes up a little bit. So what do these two cytokines do? And this is complicated. What this chart says is these two cytokines, when they're high, protect you against infection, okay? So your first line of defense when you get an infection is your effector T cells and your macrophages have to talk to each other and ramp each other up to say, let's go fight the infection. If the levels of interleukin 12 and interferon gamma are high, this crosstalk is amped up and they are more ready to fight infection than if you have low levels. So we're saying, could it be that when you have high levels of these and you're ready, you fight infection better from the start, that this helps your cognitive trajectory? And this may relate to the work that Cure Alzheimer's funded by Rob Moyer, my colleague Rob Moyer, who we all know and knew and loved, and he tragically died with a glioblastoma at 58 years old and a couple of years ago. But he championed this in my lab, the antimicrobial protection hypothesis, where he figured out that the amyloid beta protein isn't just junk in the brain. The amyloid beta protein actually gets made and deposits to trap pathogens to get into the brain, bacteria, viruses to get into the brain. The amyloid beta protein binds to them right away, traps them. It has to form a plaque to do so. So the bad news is that the microbe, small amounts of any microbe, bacteria, virus, fungus, will trigger a plaque, but the plaque is actually trapping the microbe to protect the brain. Now, we're not saying this is how all amyloid forms, because obviously we know of genetics where you can get amyloid without the need for a microbe-borne infection. But this is one way in which amyloid can form. Infection will drive amyloid, and we showed that the amyloid protects against infection. This is a mouse study. I'm not going to show all the data, but we've more recently found that infection also triggers tangles. When the tangles form in the nerve cell, either due to a virus or amyloid, the tangle blocks the virus from spreading from one nerve cell to the other across the synapse. So think about that. Amyloid, tangles, and obviously neuroinflammation, all part of your brain's innate immune system to protect you. Could it be that the reason why these cytokines that protect against infection in the blood and the outside the brain are useful on cognitive trajectory because they lead to less infection in the brain, driving Alzheimer's pathology. That's the hypothesis. And in further support of it, isn't it interesting that vaccine after vaccine is showing protection against Alzheimer's disease? Tdap vaccines, pneumonia vaccine, flu shots, even COVID vaccines. We're seeing that folks who have vaccines have more protection against Alzheimer's disease. So these are obviously vaccines hit the most common infections that do target us. And so could it be vaccines are also like those high cytokines are protecting against infection getting into the brain and driving pathology? This is new stuff. It's fringe stuff. It's the stuff the Cure Alzheimer's Fund loves to fund because it's fringe and new and exciting. And it looks like it's getting more interesting by the day. Okay, so let me keep going now. That's early detection and a little bit about infection. Let me get now into early intervention. Can I just ask the group, how much time do I have left? Because I want to gauge how fast I go and what I cover. How much time do I have left before we get to Q&A? Can someone tell me how much time I have left?
Well,
It's about 20 minutes. Okay, thank you. So as you've heard, just in June, there was an approval of the first new Alzheimer's drug in decades. And this is Aducanumab, or as it's called by Biogen, Aduhelm. And it's been controversial, right? FDA approves Alzheimer's drug despite fierce debate whether it works, controversial, Alzheimer's med, safety worries. What's happening is it's been approved, but the problem is it wasn't approved for cognition. It wasn't approved to improve memory. It was only approved to move amyloid. And it's mainly approved for a small slice of patients who are in the earliest stages of the disease, where the hope is that removing amyloid can still help. And that's still a gamble, okay, based on what I just told you. So if you look at those trials, one of the trials had mild cognitive improvement. There was some incremental improvement. One trial failed, but both trials dramatically reduced amyloid. And that's because of what Aducanumab is. It's an antibody that activates these little cells in the brain called microglia to clear amyloid. And it was inspired again by Rob Moyer's work in my lab in 2005. Rob described naturally occurring antibodies that target amyloid aggregates, autoantibodies. And what he found was the more of these autoantibodies to amyloid you had, it protected you against Alzheimer's. And then a company in Sweden called Neuroimmune found those autoantibodies we described that were protective and then mimicked them in the form of Aducanumab and then licensed that to Biogen, and that's what Aduhelm is. So Aduhelm right now is being used to treat the mildest of patients, but whether it will cause cognitive improvement is completely unclear. Certainly, if you have later stage Alzheimer's or moderate Alzheimer's, you will not, almost certainly not get improvement. And this is a very expensive drug. It's $56,000 per year, plus the cost of imaging that has to be done to make sure you don't have hemorrhages or brain swelling, because there are some safety issues here. And there are other immunotherapies on the horizon as well, from Lilly, from Promise, from others. But the problem is these immunotherapies, you have to use them preferably pre-symptomatically. It's a gamble whether they'll work at the earliest stages, and they're too expensive. So think about what I said earlier. 38 million people in this country have amyloid in their brain who would like to bring it down. You can't treat 38 million people with a drug that has safety issues that's $56,000 a year. The hope is that maybe for that small section of slice of patients that are the earliest stages, maybe they'll get benefit from clearing amyloid. But I have to say that's a gamble. We just don't know if that's going to happen. So that's the bad news, right? I mean, the even worse news is if you have someone who has a loved one with Alzheimer's disease, who doesn't get covered for this drug, and they say, well, for dad, we're going to mortgage the house to get the $56,000 to pay for this. Well, if dad has Alzheimer's that's even mild to moderate, it's not going to help. So that's important to know. The silver lining here is that because the FDA approved a drug that removes amyloid, this opens the door for other drugs that move amyloid that can get approved. other drugs that are much cheaper and safer. For example, Cure Alzheimer's Fund has been funding Steve Wagner and I to develop gamma secretase modulators. This is just a pill you would take that reduces the amyloid in the brain. Where thanks to Cure Alzheimer's Fund and the NIH, we're about to go very soon into our first clinical trials in patients. It took 20 years to get there, developing this in academia at Mass General and at UCSD, right? I'm developing through as I'll talk at the end, through our Alzheimer's in a Dish model, again, that Cure Alzheimer's funded, natural products and approved drugs that might likewise clear amyloid exactly the same way as Aduhelm. They tell the microglial cells in the brain to eat it up while you sleep. So the good news is this has opened the door for other cheaper, safer drugs, where once we show they remove amyloid, we go to the FDA and say, look, we're removing amyloid, but we don't think that's good enough to help patients who are already suffering. We got to do other things for them. We got to stop their neuroinflammation. But there are 38 million people who are going to find out through a blood test or imaging soon over the next decade or years that they have amyloid, high amyloid in their brain. And this little cheap white powder, this supplement or this repurposed drug, we can give to 38 million people safely and bring the amyloid down the way we bring cholesterol down with a statin. That's the vision going forward, that now this new approval, as controversial as it is, makes possible. So this very awkward controversial approval has also initiated a new era in Alzheimer's, where maybe we'll be able to develop amyloid drugs that are cheap and safe, that can be used as secondary prevention, the way we treat cholesterol for heart disease. How do we get there? How do we get to those cheaper versions of these drugs? Well, like I said, Cure Alzheimer's Fund was bold enough to fund our attempts in our lab with Du-Young Kim to create Alzheimer's in a Dish, and we did it. This is an index-sized tray. Every little well here is the size of a paper punch, and each one is a mini human brain organoid, where in 1 month we get plaques, the plaques cause tangles, and then you get activation of glia and neuroinflammation, the whole thing. And we can just then start screening. This may, the New York Times wrote about this in 2014, and it said, this is making drug discovery 10 times cheaper, 10 times faster. Well, guess what? It's made drug discovery 100 times cheaper and 100 times faster, as it turns out. And we've been going great guns with this with the Cure Alzheimer's Fund 3D drug screening program. 3D cell cultures are what we call this Alzheimer's in a Dish. So 3D drug screening. And the first thing we did is we said, let's target neuroinflammation. Again, thanks to the Alzheimer's Genome Project, we were the first ones to find a neuroinflammation gene in Alzheimer's in 2008. Now there are dozens of neuroinflammation genes for Alzheimer's, for late onset Alzheimer's, shown here. We found the first one. Why? Because Cure Alzheimer's Fund was the first to fund a whole genome screen in our families, and CD33 was the first target. Then Decode Genetics in Iceland found a second one, TREM2. We've found many more since then. Now there are over 100 Alzheimer's genes. I don't have time to talk about all of them, but the fastest growing group are the neuroinflammation genes. And let me tell you, when we first found CD33 and said these neuroinflammation genes might matter, we were alone. Cure Alzheimer's Fund and our lab were alone. And now it's probably the most studied part of Alzheimer's disease, in addition to amyloid. Well, what do microglial cells do? Because these genes all work in microglial cells. Microglial cells have 3 roles in the brain, okay? They're nurturers, right? In other words, they clean up debris, they help the neurons to survive. Even when you're learning new things, they're helping to sculpt your synapses so that you can learn more effectively. But they're also sentinels, okay? They're always on the lookout for something going wrong in the brain. They're looking for infection. So what happens is while you sleep, these microglial cells are eating up amyloid and clearing debris. That's why sleep is so important. But if they start eating and they say, ooh, I just ate a piece of a dead neuron, now the sentinel says, wait a minute, a neuron's dying. And now if that happens enough, they switch from being housekeeper, nurturer to being warrior, soldier. They change shape and they say, neurons are dying. They assume it's an infection and they wipe out that part of the brain to prevent the infection. Well, so think about what I told you before. Amyloid tangles, cell death, tangles are propagating and spreading, kind of like the amyloids to match, tangles are like brush fires that are spreading. But when those microglial cells get involved, now you have the forest fire, neuroinflammation. There's even the word flam and inflammation. The microglial cells activate the astrocytes. Now all the glial cells change shape and they gang up on the neurons and they kill them. Okay, they kill them in all kinds of different ways. This is what we have to stop in a patient. The match has already been lit decades ago. The brush fires have been spreading for decades. Now it's that forest fire that's taking you out. You have to either protect against it or stop the fire. And so now we have the genes that tell us how the microglial cells can go from housekeepers to assassins, which genes go up and down, and we can target those genes. So we know that if CD33, the first gene we found thanks to the Alzheimer's Genome Project of Cure Alzheimer's Fund, if CD33 is up, the microglial cells go nuts and they try to kill everything in sight. So we have already developed gene therapy in mice. We have a lot of work to bring it to humans for bringing that CD33 back down. Companies like Elector and Denali are trying to use antibodies to hit these genes. So these two genes that started with Cure Alzheimer's Fund are now the main targets for hitting neuroinflammation in AD. We wouldn't have had CD33 or the genes afterwards if it wasn't for Cure Alzheimer's Fund and the Alzheimer's Genome Project. And now, like I said, we can create all of this in a dish. Okay, I wanted to show you something really quick. These green guys are neurons, nerve cells. The blue guys are microglia. These are healthy nerve cells in a dish, peacefully coexisting with the microglia who are cleaning up after them and nurturing them, okay? Now, in the next slide, if I can get this to go, in this slide, the nerve cells were making plaques and tangles and starting to die. Look what happens. Many more microglial cells come in, and what are they doing? They're eating the nerve cells. So we can actually model this whole cascade in a dish, and then we can say, let's find drugs that stop it. And we found a bunch of drugs. These are all the drugs, 51 drugs and natural products that, just like Hydrocanumab from Biogen, get microglial cells to clear amyloid. 51 of them that we can start working on for trials. Okay, sorry about that. And then we have about 40 of them that stop the neuroinflammation. In other words, the microglial cells causing that cell death and causing neuroinflammation. You know, one of the hits, for example, is cat's claw. This is a particular type of cat's claw that comes from a company I started, that PTI 073. And what you're seeing is the more cat's claw you add, the more the microglial cells are clearing amyloid. So this is just an example. But we have all these different safe drugs and compounds now, and we can start doing the trials. We're now doing fundraising to start small trials on these drugs that can hit the neuroinflammation or take the place of aducanumab to clear amyloid. The ones that clear amyloid, we'll use those early on in 38 million people who need them. The ones that hit the cytokines, those will, and the inflammation in the brain, those will be the ones we use to try to help patients and tamp down that neuroinflammation. This is, don't worry about this slide. I just want to show you, this is how complicated our drug screening is. This shows the whole process we figured out of how Alzheimer's is caused. And then all the different red intervention points where we're screening for drugs, approved drugs and natural products that can hit this disease at different spots. And so this is what we're doing right now. And we have tons of candidates. We have 44 new drugs and natural products that stop amyloid, 31 that actually prevent the amyloid from causing tangles. We have 42 of them to stop neuroinflammation. And our next goal is to get these into clinical trials with platform trials at Mass General and elsewhere with Cure Alzheimer's Fund, and hopefully help from the NIH as well. So this is a very exciting time. Two of our drugs that came out of our screens already worked in ALS. These were drugs that afforded neuroprotection. They protect against neuroinflammation. They help the neurons to survive. They worked in ALS. They're now, and Cure Alzheimer's Fund helped this company, Amylyx, which I helped co-found 8 years ago with two young guys from Brown University. They were students. And this neuroprotective drug combination is now being considered for approval by the FDA, Canada, and Europe. And it's also being used in Alzheimer's disease in a phase 2 trial. So in other words, if you have drugs that protect against neuroinflammation, you can try them in Alzheimer's, Parkinson's, ALS, Huntington's disease, because in all those diseases, there's different ways you get the initial cell death, but then you trigger neuroinflammation that causes the big cell death that leads to the symptoms. So these two drugs, which work in the neurons in a dish, afforded neuroprotection and looked like They may be very well the newest approved drugs for ALS, and we're hoping that they might help in Alzheimer's as well. So what I've told you is that to prevent Alzheimer's, we need to stop plaques and tangles early, decades before symptoms, but to treat a patient now, it's not enough. You have to protect the neurons from dying due to neuroinflammation or stop the neuroinflammation. Now, in the last 5 minutes, I want to just quickly tell you what can we do now in terms of lifestyle and interventions. And the good news is that, look, 95% of Alzheimer's genes don't guarantee the disease, even ApoE. Lifestyle makes a difference. Lifestyle can mitigate genetic risk for dementia in 95% of cases. In those early onset cases, they need extra help. We're hoping our gamma secretase modulator I talked about earlier that Cure Alzheimer's is funding will help those early onset patients and reverse what's happening in them where they have a death sentence. And so that's really good news that we think we can help them. But in most cases where you don't have the mutations that guarantee the disease, the genetic variants just increase your risk, and lifestyle matters and can make a difference as to whether you get the disease. How can you make a difference? Well, there's Life Simple 7, blood pressure control, no smoking, regular physical activity, reduce your diabetes risk, check for diabetic factors, healthy diet, weight control, lipid control. All of this goes a long way for promoting brain health as well as heart health. And many of that is summarized here. I've tried to make it simpler by coming up with the acronym SHIELD, which stands for sleep, handling stress, interaction with others, exercise, learning new things, and diet. So let me just walk you briefly through that. It's during sleep, 7 to 8 hours a day. If you only get 6 hours of sleep, you can take a nap, okay? Every time you have dream, REM sleep, and go into deep sleep, that's one rinse cycle where you get rid of amyloid. During that time from REM to deep sleep, those little microglial cells are like scrubby bubbles. They're nurturing, they get rid of the amyloid. And they work and they do that while you're sleeping. That's why you need to try to get 7 to 8 hours a day of sleep. Stress kills nerve cells. Stress causes cortisol release, and the nerve cells die, and you start triggering everything from tangles to neuroinflammation. How do you deal with stress? First, we have to define what stress is. Stress is what the brain, in terms of the brain, stress is what the brain does when you are faced with a separation threat that's challenging your attachments. All your life, you're developing certain dependencies and attachments to things. As you get older, your security is based on what you're attached to. And you're separated from those attachments that make you feel secure. That causes stress. So if you look at stress value, right? If you look at stress values, number one is death of a spouse or life partner, then divorce, marital separation, detention in jail, death of a close relative, et cetera. These are all different degrees of separation from things in which you have a great attachment. That stress then causes problems. Chronic stress causes inflammation, and that accelerates aging. It's often called inflammation. So dealing with stress, meditation practice, being more mindful in the moment goes a long way. Interaction with others. Being socially isolated or lonely can also increase stress. It also reduces intellectual stimulation. So being lonely increases risk for Alzheimer's twofold. If you're alone and you like it, that's fine. But if you're lonely, meaning you're alone and don't like it, increases your risk. So staying social, interaction with others is important. E is exercise. Exercise is amazing. It causes the birth of new nerve cells in the hippocampus where Alzheimer's begins. The increased blood flow brings things to the brain. Like we just had a paper recently in Nature Metabolism and in Science showing that when you work out, your muscles produce something called irisin that goes into your brain and actually helps reduce inflammation and increases neurogenesis, the birth of new nerve cells in the brain. Exercise also induces an enzyme in the brain that breaks down amyloid. So exercise at many different levels. How much exercise? If it's robust exercise, like I have my recumbent bike behind me, I do a half hour every morning, 90 RPM at a resistance of 5. If you're not doing excessive exercise for a half hour, then at least get a 1 hour brisk walking every single day. Try to aim for 10,000 steps per day, but people are saying even 7,000 steps per day is good enough. L is learn new things, right? When you learn new things, you make new connections in the brain. You make new synapses. And when you make new synapses learning things, you also reinforce existing synapses, because when you learn something, you associate the new thing with the old thing. So new synapses connect to old synapses, and you're reinforcing your neural network. You're building up your synaptic reserve. So I tell people, when you retire, think about building up your synaptic reserve just like your financial reserve. The more synapses you have, the more you can lose before you lose it. So you have to build up synapses so that as age-related changes occur in the brain, as synapses start to die, right, you have more synapses to fill in the gap before you get into trouble. So building up synapses by learning new things. Brain games are fine for teaching you how to focus, and that might help you to learn better, but won't help you unless you're learning new things, hobbies, instruments, languages, doing new tasks, gardening, anything that involves learning, even reading a good nonfiction book or watching a documentary, learn new things. And finally, diet. The most important thing about diet is your gut microbiome, the bacteria that live in your gut. You have trillions of bacteria in your gut, 8,000 different strains that control everything from your mood in your brain to inflammation. We've even shown that these gut bacteria can control amyloid. Cure Alzheimer's Fund funded projects on this. The way you keep your gut microbiome healthy, which will keep your brain healthy, is Mediterranean diet, okay? Less meat, more fruits, vegetables, whole grains, seeds, nuts, etc. And this just shows a Mediterranean diet. Pyramid, a little red wine for antioxidants, a little dark chocolate for antioxidants. But most importantly, it's all about what are called probiotics and prebiotics. So some of you may take a probiotic or eat yogurt, but as important, or maybe more important, are prebiotics. That means plant-based diet, seeds, nuts, whole grains, and that type of thing. That's what your bacteria in your gut like to eat. And when you feed them well, they treat you well. That's the bottom line. When you combine probiotics with prebiotics, that's a symbiotic. When we treated Alzheimer's mice with a symbiotic, we dramatically decreased plaques and neuroinflammation in their brain via their gut. Here's a simple symbiotic for breakfast: some yogurt with live cultures and some granola. And if there's some dark chocolate as an antioxidant, even better. Very simple symbiotic you can have every day just during breakfast. This Mediterranean diet, I think you all know what that is. Add a side salad or raw veggies to your lunch or dinner. Antioxidants from blueberries, green tea, dark chocolate, red wine. Take omega-3s. If you're taking fish oil, make sure it's very clean, triple distilled. I do a vegan sourced omega-3, that's very good. And consider a probiotic supplement. I use Nordic Naturals probiotic because I have a prebiotic in it as well. I don't talk about supplements anymore in my talks that I take personally or recommend, just because of legal issues, and there's so much crap out there, to be honest, with brain supplements that don't work. But if anyone's interested in the supplements I personally take and want to know more, you can contact me directly. We've even started a new company that's going through all the gut bacteria and seeing which ones are most useful for protecting your brain. We're trying these now in mouse models, and it's actually quite good. The idea is to someday have a very customized probiotic of bacteria way beyond the ones you get in your yogurt, ones you never heard of that can protect your brain. So we're screening thousands of strains of bacteria in the gut to see which ones afford the most protection against neurodegenerative disease, neuroinflammation. So I'll conclude by saying, just remember that at any time you can start with these new habits that improve your brain health. I wrote 3 books about it. And I like to say your mind, body, and brain today are the results of your choices and habits yesterday. But your mind, body, and brain tomorrow are the results of your choices and habits today. And you can change those at any age. So I'll just summarize with the lifestyle recommendations, which are quite simply stated here. Sleep more, meditate more, meaning be more mindful, see friends more, move more, learn more, eat less and healthier. And again, because I'm a geneticist, I also have to say, choose your ancestors wisely. So I'll stop there. I'll thank Cure Alzheimer's Fund for funding this work. Thank you to the professional staff in our unit. Here's our boat trip from a couple of years ago. We're hoping that COVID will allow us to do this again someday, our collaborators. And I'll stop there and happy to take your questions. Thank you very much.
Thank you, Rudy. That was great as always. We have a number of great questions, and I encourage people to keep putting them in the chat. I am still checking as we go. and you gave us a lot to think about and a lot to ask about. The very first question that we got was actually, what can we ask of our doctors starting in our 40s so that we actually get better than a physical? We don't just get the physical.
Well, right now, the only thing that's offered, I mean, if you wanted to pay out of pocket, you could ask for the amyloid test or some of these blood tests that are FDA approved or even imaging. It's up to you whether you want to know, because the only thing you can do about it right now is a lifestyle change, right? Until these meds get on board. Of course, if you have very, very earliest stages of symptoms plus amyloid, you might qualify for Hydrocanumab and coverage. That's a very small slice of people. But you can ask your doctor otherwise whether he or she can give you a neuropsych exam. The most common that's done at a checkup is the Montreal Cognitive Assessment, or MOCA, M-O-C-A. And again, it's better than nothing. It just gives you an idea of whether you're slipping or not. It might, if you don't do well on the MOCA, it might tell you, hey, maybe I'll check to see if I have amyloid brewing in my brain with a blood test or with imaging, or look for tangles in the brain. Again, it's not actionable. We don't have the drugs online yet. We have some supplements that we can recommend that may or may not work. We have lifestyle interventions that can help but are not guaranteed. I know that out there's the Bredesen protocol. Some of you may have heard of, where Bredesen guarantees curing and reversing Alzheimer's. We would never say that. Lifestyle can help, but we can't say it's going to guarantee anything. But that's about it.
Thank you. So just getting back to the MOCA for a second, that Montreal cognitive testing, is that something that we should be asking our doctors for before we have symptoms, so we have a baseline? How often can we take that to see whether we are in fact slipping? What's useful?
Yes, that's a good idea. Having a baseline. In fact, that's why we started this React Neuro company, is that we hope that this device that does a neuropsych exam in a much more detailed way and does eye tracking and voice analysis at the same time, the algorithm spits out your brain health index. Well, we're doing sports teams right now, senior living, but our hope is that as we get this thing validated and get enough data, it will be in every doctor's office, that after you get your blood pressure checked for 90 seconds, you put on this device and you see where you're at. Well, we're getting there, but for now, it would be a good idea to baseline against something. And I think the MOCA is the best we got for just general baselining right now.
You started your talk discussing Alzheimer's as the biggest driver of dementia. How might somebody know if someone's symptoms are more likely to be dementia versus Alzheimer's disease, and what other causes might be driving cognitive decline?
Yeah. So there is no dementia versus Alzheimer's. It's dementia as the umbrella term, and the most common cause of dementia in the elderly is Alzheimer's, but there are other causes of dementia. Other causes of dementia include frontal lobe dementia, and can be having mini strokes that you don't notice, neurovascular stroke-based dementia. And actually, a good psychiatrist or neurologist who specializes in Alzheimer's will be on average 90% accurate in telling you whether it's dementia due to Alzheimer's or frontal lobe dementia or neurovascular dementia. So your PCP can give you an idea that there's an issue, but it's a neurologist or a good psychiatrist specializing, or a gerontologist specializing in the brain, that would have to tell you whether it's dementia due to Alzheimer's or frontal lobe dementia. And they're usually about 90% accurate. Like with frontal lobe dementia, it's not just memory, it's also disinhibition. You may be doing things that are unusual. You may be less restrained and inhibited, whereas normally your frontal cortex is restraining what you do, right? You may be saying things or doing things that are very unlike you personality-wise. And that might be more of a frontal lobe dementia issue than an Alzheimer's issue. So a good psychiatrist, neurologist can figure that out.
So we have a question about if the statistics about how many people will develop Alzheimer's as they age are pretty scary. If I am a person in my 80s and I'm functioning well right now, how worried do I need to be that I'm going to develop Alzheimer's disease? Oh, you might be frozen, Rudy.
Well, look at your family history. That would be the first thing to do. Oh, it's okay now?
Yeah, you're
Am I
back.
Okay. Yeah, it says I have to reboot my modem again. Thank you, Comcast. So the first thing you want to do is look at your family history, and see if see if first degree relatives had signs of Alzheimer's and at what age, and that will give you an idea of whether you might have increased risk and at what age. Other than doing that, you can also check if you have ApoE4 variant. The E4 variant will increase your risk, 1 copy by 3.7-fold, 2 copies 4-fold. But they don't guarantee you're going to get it. So that's genetically. And otherwise, just the data suggests that at 85, it's about, on average, 40% of the population that shows early symptoms of cognitive impairment. And so if you want to know if you're in that 40% of people who are going to need to worry at 85, first thing you would do, like I said, is look at your family history, maybe consider one genetic test like ApoE4 if you want to know. You can also you could pay out of pocket for imaging for plaques and tangles. You can pay out of pocket for the blood test. So there's things you can do to find out. But right now, we don't have the drugs to help you. They're in development. And the only thing you can really do is change your lifestyle according to SHIELD. Even in the Bredesen protocol, what they recommend is fine. It's just that we would never say it guarantees cure or reversal. That's just not possible. We can just say this is the best thing you can do to help yourself.
If I were that 85-year-old who is functioning really, really well and is just worried because the statistics say so many of my peers are going to develop Alzheimer's or already have, if I were scanned and there were amyloid in my brain, might I actually be a resilient patient or a resilient person because I haven't developed cognitive symptoms?
Well, yeah. There are folks who die with lots of amyloid in their brain with no symptoms. There are folks who die with lots of amyloid and tangles in their brain with no symptoms. The key is whether you triggered neuroinflammation. These new biomarkers we just found that if you carry them suggests if you have amyloid in your brain, you're still fine after 6 years versus others. We're trying to get those, more studies on those and get those eventually FDA validated to test what is your risk for dementia after you have an amyloid diagnosis in your brain by imaging. So these are all works in progress, but anybody might be resilient. It's common actually to see people with amyloid in their brain, but somehow didn't get tangles and inflammation and they're fine. It's a little less common where you see people with amyloid and tangles in their brain and no neuroinflammation. But resilience comes from having less inflammation or having more synapses to lose before you get into trouble. So that's where the learning new things comes in. That's so important.
So that actually leads perfectly into another question that we've got, which is, how do we really know that amyloid is the culprit? If it's only neuroinflammation that leads to the neurodegeneration and the cognitive symptoms, how do we know that amyloid is central here?
The genetics guarantees it. I mean, you have mutations that guarantee this disease by 60, and they're in different genes, and the only thing they have in common is amyloid and making more amyloid in the brain. And we now know from Alzheimer's in a Dish, thanks to Cure Alzheimer's Fund, amyloid directly causes tangles. The big controversy, amyloid versus tangles, for decades was because mice, where you put in the Alzheimer genes, would make amyloid but not get tangles. Well, now we know mice can't make tangles. That's why they didn't have them. Okay, so we now know that amyloid directly causes tangles, just as bad as bangs to the head. That's the first cell death that then triggers neuroinflammation. So there is absolutely no doubt that amyloid triggers this disease. It's not whether it does, it's when it does that matters. It does it decades before symptoms. That's why when you do trials on amyloid and they fail, it's too late. It's like someone needs a bypass, a coronary bypass, and you do a trial on Lipitor to see if it fixes their heart. Of course it won't. You had to take the cholesterol drug decades before. Perfectly analogous with amyloid.
Okay. We have some questions on specific therapeutic options or things that people have been told might be therapeutic options. For one, this question about probiotics, do you have to take a specific probiotic for a specific condition? Is this like a drug that you only take for that you match to a specific disease, or are they a general option?
Well, right now, that's a great question because it tells us where we're going in the future. Right now, the probiotics are pretty standardized, right? It's what you read on your yogurt container, right? Bifida, Lactobacillus, Acidophilus, et cetera. Now, what we're doing with this new company we started with Gary Ruvkin, my scientific co-founder, he discovered microRNA, which is the biggest thing in epigenetics. He'll get the Nobel Prize eventually for that. But he spent the last 10 years in his lab isolating every strain of bacteria that lives in the gut that he could possibly find using human poop, okay? So that's how they find them. There are 8,000 different strains of bacteria in your gut. On your yogurt container, there are 10 or 12. So we like to say we're screening them bugs as drugs. So we screen them to see which ones can protect against neuroinflammation and protect neurons from dying. And the idea is that you give those bacteria to the gut and they make metabolites that go to the brain and protect the brain. My guess is, to answer this question, that for different diseases, if you want to stop the end game of neuroinflammation or protect against it, it's going to be the same bacteria. Alzheimer's, Parkinson's, Huntington's, ALS, doesn't matter. The same bacteria that protect against cell death, like the Amylyx drugs, are going to work across the board if they work. But there may also be bacteria that specifically help bring down amyloid, which would be a way to prevent Alzheimer's early. And we already know that we can change amyloid levels by changing the gut bacteria. There may be different bacteria that may help bring down Lewy bodies that trigger Parkinson's disease. So that would be the next phase, is how to find specific bacteria that hit the initiating pathologies, plaques, tangles, and Alzheimer's, Lewy bodies, and Parkinson's. But the bacteria that protect you against neuroinflammation, most likely will be able to use those across the board. And they're not the bacteria on your yogurt container. Those are helpful. These are bacteria with very arcane names that you've never heard of. And right now we're dealing with the FDA because the FDA doesn't know what to do with this. Because we go to them and we say, look, we have these 6 bacteria that normally live in your gut. You just never heard of them, and they have crazy little names. But in an ALS mouse model, they have dramatic effects, and we want to do an ALS trial or an Alzheimer's trial. And the FDA scratches their head and say, well, wow, these are bacteria that you normally have, so they should be safe, but they're not the ones that people have been eating for decades in their yogurt. What do we do? So they're still trying to figure out how we do safety trials with this. So we're breaking new ground here.
So it sounds like that then ties back to your advice about diet, because if you can't take a probiotic in pill form, it sounds like your best option is to eat the healthy diet that will provide probiotics in your food.
Right, but I would argue that prebiotics are way more important.
Okay.
Probiotics, look, you got trillions of bacteria in your gut. You're adding what, 1 billion? It's like you have a fish tank you want to keep healthy, and every once in a while you add a couple more fish, as opposed to feeding them, right? The best way to deal with your gut microbiome is to feed them plant-based diet, seeds, nuts, whole grains, fruits, cruciferous vegetables, right? Anything plant-based and crunchy is going to make your gut microbiome happy, make it balanced. That will help mood, and that will also help with inflammation in your brain. This isn't woo-woo, it's not new age science, it's absolutely documented work.
Okay. I'm going to ask you another couple questions before I turn to Bruce and Jeff to see if they have any questions. But I'm sorry, we're not going to be able to get to everybody's questions because we're getting great ones. We did have another question about two more questions about specific options that people have seen recommendations for. Can you talk about whether there is solid scientific evidence for benefit from CBD or L-serine?
L-serine, absolutely not. Sorry. There are no data of any value, in my opinion, as a scientist, to support L-serine, including the animal data. It's flawed. Sorry. CBD is a work in progress. CBD will help curb neuroinflammation. And the CBD, and you don't need THC, so you don't need to get high. But CBD that's THC free does have some anti-neuroinflammatory properties. The question is, how well does it get into the brain if you're taking a CBD oil, and which oil is the best? And I think the jury's still out on that, but there's potential hope for CBD.
Okay, so we still need more data, but it's an ongoing story.
Mhm.
All right, well, if I can ask Bruce and Jeff if they have any questions.
Jeff, you're muted.
And Bruce, I think you had a couple and you're muted as well.
Well, I'll go ahead with one. I have a lot of friends that have Parkinson's disease, and we know that Parkinson's will morph into Alzheimer's. Can you describe, Rudy, what happens when somebody with Parkinson's ends up with Alzheimer's disease?
Mhm.
What the cause is, and is there anything that people with Parkinson's can do to prevent that?
Yeah. So, and I'm glad you asked that because I didn't mention this, that when you talk about plaques leading to tangles, leading to inflammation, inflammation can then drive plaques and tangles. So it's a vicious cycle. So in Parkinson's disease, where you have a different part of the brain involved with movement, you have a protein called alpha-synuclein forming Lewy bodies. Same thing in Lewy body dementia. The Lewy bodies are just like tangles, they propagate and spread. And as they spread, they can move to different brain regions. And that can trigger cell death and inflammation. And if that happens in the hippocampus, that will give you Alzheimer type symptoms. But the inflammation and what's called oxidative damage that comes from inflammation can cause tangles and even amyloid to form. So you can have one disease pathology like Lewy bodies through inflammation spreading to another region, induce pathology of a different type of disease like Alzheimer's. So the way it would go is Lewy bodies, inflammation, Lewy bodies spreading, inflammation spreading. Now you get to the hippocampus, and the inflammation is causing the birth of new plaques and tangles and causing more inflammation. So you see mixed pathologies. But from a symptomatic point of view, the main reason why you can see Parkinson's patients later getting dementia is because the Lewy bodies have spread to the hippocampus and cortex, driving inflammation there. And along with that, you might also see some plaques and tangles.
Is there anything somebody with Parkinson's can do to stop that?
Same as Alzheimer's, whether it's Parkinson's, ALS, or Alzheimer's, Huntington's, frontal lobe dementia. There's proteinopathies, misfolded proteins that trigger the disease: amyloid, alpha-synuclein, tau, Huntington, TDP43 and ALS, SOD1. Those misfolded proteins kill the initial nerve cells in some part of the brain or central nervous system that gets the ball rolling. But then in each case, you pyramid up to neuroinflammation, and that does the bulk of the killing that causes the symptoms. So if the bulk of the killing is in a part of the brain for movement, you get Parkinson's, or motor neurons, you get ALS, or pyramidal neurons in the hippocampus, you get Alzheimer's disease. So again, for patients who are suffering with symptoms of any neurodegenerative disease, if you want to prevent it, hit the proteinopathies to misfold the proteins early, plaques, tangles, Lewy bodies, decades before. If you want to help the patient now, stop the neuroinflammation or prevent the neurons from dying. That's why I'm excited by the company Emalex that Cure Alzheimer's funded, because we have a drug combination that does protect against neuroinflammation and ALS. It's probably there's a good chance it's going to be approved for ALS. And now we're hoping it might work for Alzheimer's and other diseases too. Same thing with the gut bacteria. If we get results in ALS, we go to Alzheimer's. So anything that helps neuroinflammation to go down or stops the neurons from dying from neuroinflammation, which by the way will also tamp down neuroinflammation because you feed it less. You can use those, you can try those across the board of all neurodegenerative diseases that come with aging, Parkinson's, Alzheimer's, et cetera.
So we know that CD33 accelerates inflammation, and we know that TREM2 protects against it. And we're working on ways to jack up the TREM2 and jack down the CD33. I presume any neurological disease where somebody is in the inflammatory stage would benefit from drugs that affect CD33 and TREM2. Is that right?
I agree. Yes, you're a great
Yeah.
student, Jeff. And I would say that the only caveat is I would much rather turn down CD33 than turn up TREM2. Because when you turn up TREM2, it does help with inflammation, but there is some data from David Holtzman, who's the co-chair of our research leadership group with me and Bob Vassar, that shows that when TREM2 is up, you might also accelerate the spreading of tangles. So it's not as clean a strategy as tamping down CD33. You know, we successfully carried out gene therapy for CD33 in mice, and that was very difficult to do. Microglial cells are very difficult targets for gene therapy, as it turns out. And there are companies out there that are using antibodies against CD33 to tamp it down. I'm much more excited about those prospects than companies that are trying to activate TREM2, just because there might be side effects of that.
Thank you.
Well,
Bruce?
Rudy, you had two charts up at one point that sort of showed decline. And my question is, if you were to go do, let's say you did the MoCA test, how often should you take it to get a trend, a baseline and then a trend? And then what's the rate of decline that's just natural aging versus the beginning of dementia or Alzheimer's?
yeah, it differs for different people based on genetics and lifestyle. So it's, the average would be a very incremental decrease in performance on MoCA. But if your lifestyle is really bad, you're not getting enough sleep, or let's say 1 year your MoCA is pretty good, then that coming year you eat junk food, you're not getting sleep, you're not exercising, you're stressed out, you're a couch potato, your MoCA might go down considerably after 1 year. So it really depends on both your lifestyle and your genetics. That's what's going to determine how fast you just decline as part of age. Some folks, can turn it around. I mean, you might be declining a little bit, and next thing you know, you insist on 7 to 8 hours of sleep. I get 7 to 8 hours of sleep on weekdays. On weekends, I sleep 9 hours. Okay, Tom Brady sleeps 9 hours every day, and look how he's doing. Diet. I'm a vegetarian, plant-based diet. I try to do everything I can to try to keep my brain not just healthy, but I want to keep my brain optimized. I want to maximize the potential of my brain. That's why I insist on SHIELD to the max. So, I would say you begin baselining, I would say, especially if you're over, I would say by 50 for most people, unless you have a family history of dementia. But by 50, just stop baseline, ask your PCP or have them refer you to a neurologist or psychiatrist, stop baselining where you're at, as Meg had suggested.
One other follow-up. I find that the field of medicine doesn't deal with alcohol very often. It tends to be a quiet factor. People eliminate salt, they eliminate sugar, extra added. What about alcohol? Would it be helpful to stop drinking altogether?
Well, alcohol is a double-edged sword, right? Moderate alcohol helps with neurovascular conditions. So blood thinning, your baby aspirin, a little bit of alcohol. Red wine has flavonoids and resveratrol that's also helpful as an antioxidant. So a little bit of alcohol, especially red wine, is probably useful. Now, on the other hand, alcohol is killing neurons in your brain. There's no doubt that alcohol kills neurons. But from what we see, that would be triggering some neuroinflammation, but it's not triggering plaques and tangles. And the reason why that's good to hear is that plaques and tangles spread. They're propagating pathologies. Neuroinflammation is not a propagating pathology. It happens where it is, and it will keep ramping up where it is, but it's not a spreading pathology. So the inflammation due to cell death due to alcohol will be localized. It depends on how much you drink. But you don't see neurodegenerative disease pathology coming out of alcohol drinking. Now, for severe alcohol drinking, alcoholics drinking way too much, there's a dementia that's alcohol induced because you're killing so many neurons with alcohol and causing inflammation. You get a dementia just due to that alone, but there's no plaques or tangles. It's different from Alzheimer's disease. So I'd say drinking, in moderation could be, some studies say it's beneficial, especially red wine, because of the positive effects on your cardiovascular, neurovascular blood flow system. And that offsets the risk due to some of the cells that die, especially if you're not drinking a whole lot.
Okay, thank you.
So Rudy, I'm going to ask one last question that gets us actually back to the first question. But I realize that we're getting close to 90 minutes and I want to respect your time as well as that of our
I'm
viewers.
fine. As long- You guys, I don't have to go anywhere right now, so it's up to you.
Okay. You've talked a lot today about neuroinflammation and how important it is. And you showed us some images of scanning for tau and for amyloid in our brains. How can we know what our neuroinflammation looks like? Do blood tests for inflammation tell us anything? How can we find out more about that aspect of our brain health?
Yeah, it's a truly unmet medical need. We do not have brain imaging for neuroinflammation. It's very subpar. It's not very good. Most people wouldn't even recommend it. We're way behind on neuroinflammation. Why? Because we didn't really pay attention to neuroinflammation in Alzheimer's, frankly, until Cure Alzheimer's Fund, and I'm serious, funded the Alzheimer's Genome Project, and that allowed us to find CD33 as the first neuroinflammation gene in Alzheimer's, the first gene that indicated innate immunity. And then now that's exploded. But we started it with Cure Alzheimer's Fund taking a risk. And so neuroinflammation and the genes involved, we've only been studying now for just over 10 years, whereas we were studying amyloid since we found that gene in the mid 80s, right? So neuroinflammation has to catch up. The fact is we don't have a good blood test for whether microglial cells are activated. We do have a blood test for whether the astrocytes, astrogliosis, which is downstream of microglia, are activated. So the microglial cells get activated, then they excite the astrocytes to join the damage. That's called astrogliosis. We have a marker in the blood called GFAP and another one called UCHL1 that tell you if you have astrogliosis. We don't have any markers for that first event of microglial activation in the blood, and we have very little, if anything, in the brain. What you can do is you can look at evidence for neuronal damage in the blood or neurodegeneration. There's a marker called neurofilament light chain, NFL, that tells you if neurons are dying. And that's a good surrogate for neuroinflammation, because if there's neuroinflammation, more neurons die, and then you'll see more of this NFL that's being leaked out of the dead neurons into the blood. So that's all we have right now.
Well, if you do have another couple minutes, I will keep going for just another couple minutes, but I do want to be sensitive to where we are. We have a question about brain infection, because you've talked a lot about infection today as potentially being a trigger for pathology. Do we know if early brain infections like meningitis lead to the buildup of amyloid plaques and more tangles earlier and in greater degree than would have happened otherwise?
That's a ridiculously good question. And anecdotally, yes, we do. So there have been cases, for example, out of WashU, St. Louis, that Dave Holzman has presented, that you might have, so amyloid angiopathy, meaning amyloid around blood vessels. The other place you get amyloid is around the brain blood vessels. It's called amyloid angiopathy. That's a very rare condition. Now, in Alzheimer's patients, after you get the plaques and tangles, you will see amyloid around blood vessels, but it's very rare to see beta amyloid of Alzheimer's type around blood vessels in young people. There's a rare genetic condition that might do that. So if you do see amyloid around blood vessels in a young person, you have to ask, how was it genetic? And if it's not genetic, it becomes a mystery. Well, there were cases, anecdotal cases, where young people in their 20s and 30s had severe meningitis, and then a year or two later, it was suddenly found with abnormally with high amounts of amyloid around their blood vessels of their brain. And that would suggest, since that's so rare, that the amyloid around their blood vessels at 30 years old was a result of the meningitis, where the bacteria were probably being fought by the amyloid beta protein the A beta is binding to them and trapping them by making amyloid around the vessels. Right now, it's anecdotal cases. There hasn't been a big enough study done epidemiologically, but we're trying to do that now. But at least there's some evidence to support that idea. Also, the bigger evidence comes from HIV. Okay, so in HIV patients, a big problem, even those who are being treated with HIV drugs, is HIV dementia, because the HIV virus gets into the brain and triggers amyloid. So it's very common to see amyloid deposition and then subsequent Alzheimer's pathology in HIV patients where the virus is triggering the amyloid deposition. So that's more direct evidence.
What about COVID?
So COVID would technically trigger amyloid, but the good news about COVID is that once it gets into the brain, the receptor for COVID that's needed to propagate it isn't there. So in the brain, you don't have ACE2 receptor very abundant that COVID needs to bind to get into a cell to replicate and make more of itself. So unlike herpes virus and other viruses that can replicate in the brain, COVID cannot. So COVID is mainly causing problems in the blood vessels around the brain, where we get back to the last question, is it possible that COVID around blood vessels in long haulers or COVID patients is going to trigger amyloid angiopathy around the blood vessels? And that's something we don't know yet, but we're looking for it.
So we all see commercials for Prevagen regularly, making big promises about its benefits for brain health. We are also, of course, seeing the news about the FDA pursuing enforcement against Prevagen for making those claims. Can you talk about whether there is any scientific evidence for benefit for Prevagen for brain health in general or for Alzheimer's in particular?
I gotta tell you, why a jellyfish protein that makes them glow would possibly help an Alzheimer's disease. I can make up a story for anything. I'm a pretty hypersynaptic guy who can kind of freely associate, kind of like Jeff, and come up with ideas around anything. I can't even come up with one for that. I have no idea how possibly a jellyfish protein that makes them glow is going to possibly help the brain. And I would also say that, if it helps people, fine, rock on, but I don't see how. I would also say be careful, because Prevagen protein is a recombinant protein that they're making in bacteria, and it's not regulated in this case by the FDA. So I just personally worry about, whether, I don't know enough about the quality control around making a recombinant protein in E. coli and whether, that can cause some issues in the GI. So I don't know of evidence for that. I'm just speculating here to say, be careful with that too, because it's not a regulated recombinant protein like most real biological drugs are.
Well, and there have been enforcement activities by the government for their manufacturing issues as well. So it's a complicated story. And with that, I want to thank everyone who watched today. I want to thank the PharmNet community in particular for inviting us to be a part of your ongoing discussion series. We welcome feedback. We would love to hear from you in the future. I would like to thank, in particular, Bruce Ekman for making this possible today, and of course, our wonderful founder, Jeff Morby, and our fantastic speaker, Dr. Rudy Tanzi.
Thank you.
Thank you all.